All right, we're going to dive into proteins, enzymes, and lipids. To start with proteins, we're going to be looking at the structure like we did with our carbohydrates. Same thing with lipids. We're going to be looking at the different types of lipids. We'll also dive into a little bit of like what foods are high in the healthy lipids and what foods are high in the unhealthy lipids. And this will, of course, tie in with our heart health public service announcement project that we have been dabbling in kind of off and on, and we're going to finish up here very shortly. So let's start with proteins. One of our macromolecules, remember the monomer of a protein is an amino acid. All right, so the very first thing that we need to look at, we know that the monomer is an amino acid, but like we did with glucose, which was our solid monomer example for carbohydrates, we're going to be taking a look at the structure of an amino acid. All right, and for this structure, there are going to be some basic parts. All right, so we're going to identify what are the basic parts that are going to be surrounded by a central carbon atom. So of course, amino acids and proteins are organic. Just a quick reminder, we said that for something to be organic, it must have carbon in it, but it also must be a part of the makeup of a living thing. And of course, proteins are a part of the makeup of a living thing. So now let's take a look at the different parts of this amino acid, of the different groups that are surrounding this central carbon atom. The first part that's going to be surrounding this central carbon atom is an NH2, a nitrogen that is bonded to two other hydrogens. This is an amine group. This is actually how we start to get the name amino acid, is from the amine group. So if we're on a multiple choice type question and we're trying to pick out a amino acid out of a list of carbohydrates, lipids, proteins, nucleic acids, we would see for protein or amino acid that there's going to be an NH group on the side. We will draw out the amino acid structure shortly, but first I want to make sure that we understand these four basic parts that surround the carbon atom. Next is going to be a carboxyl group, a carbon, an oxygen, another oxygen, and a hydrogen. In this group, one of the oxygens will be double bonded to the carbon, and then we have our OH, so we will draw that here shortly. So the second of the four groups is the carboxyl group. The last two are a simple hydrogen atom, which will be all by itself at the top of an amino acid, and then at the bottom of an amino acid, we're going to have what we call an R group, which is a variable chain or group of atoms, meaning that it could be any different group of atoms. As we take a look at an amino acid, the first three, we are going to find exactly the same on every one of the 20 amino acids. Okay, so we'll say this is the same for every one of the 20 amino acids. So every single amino acid is going to have that exact NH2 on the side, the amine group, the carboxyl group on the right side, and a lone hydrogen atom at the top. What makes the 20 different amino acids, and there are 20 exactly in nature, the difference between the 20 amino acids is this variable chain or group of atoms on the bottom of an amino acid. So make sure you have these four parts down for the structure of the amino acid. And now let's go ahead and draw an amino acid. And we're going to start with that central carbon. Remember, carbon can bond to four things. So this is how we start our amino acid structure. And then at the top, we have a lone hydrogen. That's the lone hydrogen that we mentioned up here. On the left-hand side is my NH2 group. On the right-hand side is my carboxyl group. Okay. Again, that's going to be double bonded to one hydrogen and then bonded to an OH. And then on the bottom is going to be the R group, or the variable group, that gives each one its specific name. All right, so every amino acid has a hydrogen at the top. Every amino acid has an amine group on the side. Every amino acid has a carboxyl group on the other side. And every amino acid then has this variable group on the bottom, which is the R group. And then that's going to be just any group of atoms. All right. For freshman biology, we just need to know this structure, need to be able to recreate this structure, and need to make sure that we understand overall the names of each one of these parts of this amino acid group. All right, so just like with carbohydrates, we're going to have a condensation or dehydration synthesis that's going to, and I'm going to simplify an amino acid with a circle. So we're going to say that AA stands for amino acid. So I have one amino acid here, right? And then we would have a condensation reaction where water is going to come out, all right? And you can see here we have a spot for that, so the water comes out. Here's another amino acid, all right? So this water comes out. And then we do need to know the name of the bond that bonds two amino acids together, okay, in this condensation or dehydration reaction, and that's going to be a peptide bond. Okay, so we're going to then have another amino acid, and again this is a peptide bond right here. Okay, so our hierarchy here goes from, we have one amino acid, then we would have multiple amino acids, which is called a polypeptide chain. And again, the chemical reaction that's happening here is condensation or dehydration. Okay, and then from there we're going to have amino acids kind of folding over each other, forming proteins. Okay. Alrighty. So as these amino acids form into polypeptides, then those long polypeptide chains are going to fold over one another. Now we're going to see protein structure. Okay, so basically the first thing here is the idea that the order of the amino acids will determine the protein structure. All right, the order of the amino acids will determine the protein structure. All right, and protein structure is going to have different layers or levels. The first level of protein structure is going to be primary structure. All right, and primary structure is simply just one long chain, kind of a flat chain of amino acids. My second structure is going to be secondary structure. Okay, this is going to form what we call an alpha helice or an alpha helix. Okay, so alpha helices, and these alpha helices, they simply occur. So if we look at our primary structure, it's just really going to be kind of like more of a long straightish chain of amino acids, and then secondary structure is going to have two forms, either an alpha helice, which is when it forms similar to like the DNA double helix, except it's just one helice, okay, or one helix. And then my second example here is going to be what we call a beta pleated sheet. So shorthand here, this would be the alpha helice or alpha helices, and then here we're going to have a beta pleated sheet, which is just going to be a flatter protein structure. We can write that like this, beta pleated. All right, and then again, what that means is that we have amino acids that are in more of a flat structure. So these are going to be more of kind of a straight arrow type shape, as opposed to the alpha helices then, which take on that helix structure. All right. Then the third structure, tertiary. Basically think of this, guys, as the proteins are just folding over each other. So you can see here, it's a good graphic for you to look at. So in this one, you see my individual protein primary structure right here that we just learned at the beginning, right? That's my full amino acid structure, no different than the full glucose structure, right? And then I'd use a circle, or we used a ball of Play-Doh to represent a glucose molecule, and you could use a ball of Play-Doh to represent an amino acid molecule as well. So now I have one individual amino acid, right? And then I have two. Okay, the bond right here is my peptide bond. Okay, so that is just the general structure, but what that's going to look like, this is my primary structure, which is going to be my individual amino acids, just a flat chain, a polypeptide chain. Then I have my alpha helix, right, which is more of a helix type shape. And then I have my beta pleated sheet. So you need to know that for secondary protein structure, it's either going to be an alpha helix or it's going to be a beta pleated sheet. And then my tertiary structure over here, we're starting to see both alpha helices and beta pleated sheets kind of folding together to form a three-dimensional structure. That's really the big idea for tertiary structures, that it is three-dimensional. From the Enzyme Virtual Lab, you should have gotten some of the basic terminology down for enzymes, but let's take a look here for enzymes and substrates. Okay, so for enzymes, basically an enzyme is anything that's going to typically help to move a chemical reaction forward. All right, so we're going to need some type of a substrate for this to happen. All right, overall though, an enzyme is going to be a globular protein, so it's usually kind of just like it sounds like, just kind of like a big glob. All right, and an enzyme is also what we call a biological catalyst. All right, because it can help to speed up the rate of a chemical reaction. All right, it's going to speed up the rate of a chemical reaction simply by lowering the activation energy. All right, lower the activation energy. And activation energy simply is the amount of energy that's needed. I'm sorry, guys. It's the amount of energy that's needed to start a chemical. All right, so there we go with our definition of activation energy. So an enzyme is simply going to lower that activation energy so it doesn't require as much energy to get a chemical reaction going. All right, and then for enzymes and substrates, how they work together, let's go ahead and draw that out. So we've got enzymes and substrates. Go ahead and I'll draw you an enzyme and a substrate. Okay, so let's say that this is our enzyme in orange. Okay, and then we'll go for our substrate in red, maybe it would look something like this. So this is my substrate. Okay, and then we're going to move to the next step. And they are going to bond, and then we're going to call this the lock and key of enzyme substrate specificity. And you should have also taken notes from the virtual lab that we went over in the last class, and that you would have gone over for homework. So there again is my my enzyme, and then my substrate fits perfectly. Okay, so whatever chemical reaction is going to take place here now is taking place, whether it's digestion, or maybe some type of a hydrolysis reaction, or maybe it is bringing amino acids together to form a polypeptide chain and then eventually form something larger. Okay, and so the enzyme is going to stay in the same shape, if you will. So here's my enzyme still, and then now I might have whatever my products are. Okay, and we'll call those the reaction products up here, right? And then I have my enzyme still.